Thank you for joining us for this ECIS webinar dealing with barrier function assays. My name is Charlie Keese, and I'll be your host for this approximately 30-minute presentation. Let me begin by explaining ECIS a bit. The idea of monitoring cells electrically came about a number of years ago when Ivor Gaver and I were at the General Electric Research and Development Center. There we realized it was possible to monitor the activities of cells, in particular their morphology, by growing them on gold electrodes and monitoring the impedance by applying a very weak AC signal. The heart of the measurement, of course, will be the electrodes that are in tissue culture wells. Here we're looking at an 8-well array you can see in the upper left-hand panel. In the center of each well is a small 250 micrometer dot, which is the active electrode. A counter electrode labeled CE completes the circuit. Of course, this is a very small electrode, but it can monitor anywhere from a single cell up to a confluent layer where maybe 50 to 100 cells can crowd onto this electrode. Of course, in many measurements, we want to look at a great deal more cells, as is the case in this webinar dealing with barrier function. And so we have a variety of different electrode arrays with additional area that allow you to monitor up to several thousand cells and give good statistical sampling. Regardless of the type of array, though, the measurement is much the same. Tissue culture serves as the electrolyte. A very weak AC signal is applied such that the measurement is non-invasive. The cells are unaware they're being monitored. This can be applied at different AC frequencies, and we apply a constant current. We monitor the voltage. By taking the ratio of the voltage and current, we come up with the impedance measured in ohms. This is Ohm's law for an AC signal, where impedance is represented by the letter Z. But the measurement is actually quite a bit richer than simply applying this very simple formula, because instead of monitoring Z, if instead we follow the dynamics, the time course changes in the current and the voltage, we see something more involved. We apply a current, as shown in this graph on the left-hand side, by the dotted red line. The voltage is measured, that's the solid black line, and of course the voltage and the current will have the same frequency. But what's important is they don't peak at the same time. Notice the voltage lags the current by amount that's indicated by the Greek letter phi. This phase difference, along with the amplitude, can be used to return the complex impedance, which we represent as a resistor and capacitor in series. And again, we can change the frequency of this measurement, and that will alter what we measure. Here's typical data measured at 4,000 hertz when we inoculate, in this case, one of these small electrodes with cells at the blue arrow. As the cells attach and spread, they interfere with the current flow. We see a dramatic change in the resistive portion of the impedance. We can see that the BSC1 cells reach confluence sooner than the NRK cells. But also there are fluctuations in the curve that are biological in nature, because notice at the base of this curve we have a trace where no cells are added and we simply have medium in the well. These curves will depend upon the frequency that we choose, but this is the typical sort of time course data that we can record growing cells on electrodes. The reason we see the changes is indicated in this next slide. The current is carried by ions, and the electrodes are defined by insulating films. As the cells come down and attach and spread, most of them may end up on the insulating films, but some end up on the gold electrodes. The current is maintained constant, and if one were to consider how the current finds its way through the cell layer, we see that in this final panel, where the solid arrows indicate current that flows in the solution path under and between the cells, whereas the dotted line indicates that some current can couple capacitively through the insulating cell membranes. Which path is taken depends very much on the current. But what we're interested in monitoring today is the problem of current getting between the adjacent cells through the paracellular space that constitutes the barrier function of the cell layer. So let's start thinking about making these measurements using electrical current to see the barrier function of cell monolayers. Again, we're going to be looking at both epithelial and endothelial cells that are known to have adherens and tight junctions, making these paths between the cells difficult for current or other molecules to flow in that space. Again, in this other cartoon version of an epithelial cell, we see the tight junctions indicated. And again, what we're interested in measuring is the difficulty of current getting through that space between the cells. We'll come back to this diagram because there are other things we might want to consider near the end of the webinar. Now, there are 4 different approaches I'm going to describe today to monitor barrier function. One is the use of impedance, simple impedance, where we don't worry about phase difference, and we're going to grow the cells on solid substrates, gold electrodes. We're also going to look at monitoring tear, or the barrier function of the cell layer, by growing cells on porated filters, which are commonly used for these measurements. Next, we'll consider looking at, instead of just simple impedance, the complex impedance, where we can model the data and say a little bit more about this paracellular space. And finally, I'm going to mention the use of tortuous path instead of porated filters and why that may make a difference in what you measure when you study your barrier function using different cell lines. So let's first consider monitoring barrier function without the use of filters, just using the ECIS arrays. Again, the important thing here is going to be the frequency that we choose. At high frequency, most of the current will be coupling through the membranes indicated by the green lines. This is capacitive coupling through the insulating membranes. The path that we're interested in, the paracellular space, is very small when we look at high frequency. But if we drop the frequency down, the ability of the current to couple through the cell membranes capacitively becomes much more difficult. And at, say, 75 hertz, almost all of the current instead will choose to flow through that paracellular space, coming out from beneath the cell and flowing around the adhesion plaques. So let's look at these high and low frequencies and what we see in this case to look at cell spreading and barrier function. At high frequency, we're going to monitor here the capacitance. As the cells come down and block the gold electrode with their membranes, this tends to bring about a drop in capacitance. And what we're seeing in this curve is the cells going from an open electrode where they're just drifting down and sit there in a rounded fashion to a fully spread confluent layer that takes maybe 4 hours to form with these MDCK cells. The green and black traces are where, in fact, we've only allowed one divalent metal ion to be present, either calcium in green or magnesium, the black traces, looking at these 4 wells. And we see that essentially the cells are able to attach and spread, and there are only subtle differences in the traces depending on the divalent metal ion of choice. Let's look at these same 4 wells, but change the frequency to a low frequency. In this case, we're going to look at 4,000 hertz and look at the simple impedance. Notice here, the same wells give very different results at this lower frequency. When only magnesium is present, the black lines, the curves basically don't come off their starting values, but when calcium is present as the only divalent metal ion, there's a striking increase that peaks maybe 10 hours post-inoculation as the tight junctions form and the barrier function is established. So again, Low frequency is the key to looking at barrier function. Let's turn and think a little bit about filter measurements. And in particular, I'm going to introduce our new instrument called the Tier 24, which monitors 24 membrane inserts in a continuous fashion. Again, to make tier measurements, we're going to grow cells on porated filters in this case, where the pore size is 0.4 microns. This is commonly used to do barrier function. In the cells sit in an inner well. In the outer well, there's culture medium, and so the cells are being fed from both sides, something that may also enhance the barrier function compared with a solid substrate, where the cells are essentially not getting nutrients from the basal side. Electrodes are placed above and below the membrane insert. We know the area of the filter. We monitor the change in resistance coming from the cell layer. And by taking the product of those two, we get the tier value in ohm centimeters squared. That's the common way membrane inserts are being used. Now, when we do the measurement, we do it somewhat differently. It depends on the type of apparatus. Here's how the Tier 24 works. Remember, when we measure the RC values, we are treating it as though it's coming from a series circuit. But the real better way to model the cells is shown here with a parallel circuit, where we have the tier, the resistive portion of the impedance, in parallel with the membrane capacitance. And so to make the measurement with the Tier 24, just briefly how that's done, we monitor the resistive portion of the impedance at a low frequency, 75 hertz. We have previously monitored the cell-free filter, and that resistance is subtracted from what we measure with cells to get the resistance coming simply from the cells. But again, because we're monitoring the series RC circuit, next we mathematically change this as though it's coming from a parallel RC circuit as we've shown above, and by multiplying that resistance by the filter area, we get the tier value. The Tier 24 instrument that we're looking here at the GUI can follow up to 24 membrane inserts. Here we're looking at 5 or 6. 2 of them have been inoculated with very heavy inocula of MDCK cells, 2 with weaker inoculations. We see this traditional peak, which is seen with these cells before the final barrier function of about 60 or 70 ohm centimeters squared is achieved. And the bottom traces are where we simply have filters with just medium on them, no cell layer. Again, we want to compare these with the more common method that's out there to monitor barrier function, and this is coming from the World Precision instruments, where barrier function is either measured using this chopstick device or what they refer to as an endome, where we have a little more uniform current flow through the cell layer. In both of these cases, there are silver chloride electrodes involved, and so these measurements are not made continuously, as that material, as I understand it, would be toxic to the cell layers in time. So instead of using this, again, we're using gold electrodes, AC signals at 75 hertz, and again, we're seeing here some more data from MDCK cells growing on filters. What we did here is at this point in the experiment, we took our membrane inserts out of the Tier 24 instrument, placed it in the endome chamber, and monitored their barrier function using the method suggested by World Precision. And we see we're getting essentially the same values with the 2 different methods, just as a means of comparison. So that's filter measurements with the Tier 24 and pore-rated filters. Next, let's think about monitoring complex impedance, where we break out the resistance and capacitance. And these measurements have traditionally been made at 4,000 hertz. You'll find many cases in the literature where resistance at 4,000 hertz has been used as a means to study relative changes in the barrier function of endothelial cells. A lot of this early work was done by either Skip Garcia or Azrar Malik, who is in Chicago. Here we're looking at some early data from Skip Garcia's lab where he showed that sphingosine 1-phosphate can enhance the barrier function of these different microvascular endothelial cells that are seen here. We're looking at the dose response. The addition of the compound is to a confluent layer. The resistance has been divided by its starting value, and so that's referred to as normalized resistance. The Garcia labs continue to use ECIS. If you go to our website, we have now a long list of publications where barrier function has been monitored with ECIS. And you notice this one, which was published in 2019, features one of the authors is Skip Garcia, so that work continues to go on. Here's a look at looking at the blood-brain barrier, again using 4,000 hertz and looking at the resistive portion of the impedance. What we're seeing here are human brain microvascular endothelial cells that are grown either alone or in co-culture with parasites and being exposed to glucose or other advanced glycation end products, such as methyl glyoxyl, the dialdehyde. And again, this is simply pointing out that 4,000 hertz resistance is widely used to look at changes in TIR values. I want you to consider one other aspect of this measurement, which I'm calling here the contribution of alpha. So let's go back to that cartoon version of the epithelial cell that I showed earlier. Again, growing in this case on a polycarbonate filter where the pores, these 0.4 micron pores, come up vertically through the filter plane, and the cells are growing on those. Now, in this diagram, I just show a few pores, but in most 0.4 micron polycarbonate filters, there may be 1,000 pores beneath each cell. Whereas we want to measure the barrier function, the problem of going through the paracellular space, we could have a complication because the current to get to that space, or the compounds, radio-labeled compounds, for example, have to move up through the filters, and most of those pores are not beneath the barrier function area, but rather deep underneath the cell layer, between the substrate and the basal membrane. And so the electrical current, or whatever you're using to monitor barrier function, has to flow in this space to get to the paracellular space. If the barrier function is high, as it is in most epithelial cells with tight junctions, you can pretty much ignore this space. But when you're dealing with endothelial cells with weak barrier function, this contribution of this path under the cell can be significant. Fortunately, there's a model that we published a number of years ago that will let you separate these two. But before I get to that, let me just point out the use of tortuous path filters. We see a picture, an electron micrograph of them. Instead of having these vertical pores, of course, it's a tortuous path of fibers, and it's very different in how the material then flows through the filter to get to the paracellular space. On the left-hand side, you see a pore-rated filter, and the red lines indicate that the current has to come up through these pores, flow under the cell to get to that space. Whereas on the right-hand side, with a tortuous path filter, the current choosing the path of least resistance simply will move through that filter, offering very little resistance to find the paracellular space, and doesn't get in that space beneath the cell. And so there is no alpha contribution. And in fact, if you grow cells using our tier 24 in this case on pore-rated filters and on collagen-coated tortuous path filters, these are the data we see with this very tight MDCK cell layer. Again, with a tight layer, we don't have to worry about alpha as these curves come to the final value of maybe 60 ohm centimeters squared. But notice with tortuous path, we lose that large peak that comes about when you use the 0.4 micron pore-rated filters, suggesting that peak is due to the fact that the pores are actually blocked before the cells come up on their adhesion plaques, accounting for that peak. That's something we want to verify more, but that's what these data would suggest. So let me get back to the ability to use a model to separate alpha and RB. We published this model a number of years ago, and it is shown that by taking ECIS data on solid substrates at different frequencies and monitoring the complex impedance, you can separate out the barrier function, RB, the problem of getting between the cells, from alpha, the difficulty of current flowing in the cleft beneath the cells. And also this will return information regarding the combined apical and basal membrane capacitance because, again, some current will be choosing this route. Now the model you can look up if you're mathematically inclined, but if you're using ECIS software, it's not necessary. You simply, by clicking with a mouse, can ask for time course changes in these three parameters instead of looking at time course changes in resistance or capacitance. It's always available to you. And here are different values that are seen using ECIS where we're looking at RB membrane capacitance and alpha. The important point here is that when we look at MDCK cells, the dark blue rows, we see they have barrier functions depending on the type of cell from around 100 or 88 in this case up to several thousand ohm centimeters squared. But look at HUVEX, only on average about 3 ohm centimeters squared. In that case, with the barrier function that low, the contribution of alpha has to be considered. Let me show you some data with HUVEX. In this case, they've been grown to confluence, and then we've added thrombin, and it will bind to a protease activated receptor and cause this drop in the resistive portion of the impedance measured at 4,000 hertz. We can take these same data again and ask to see changes in the model parameters instead. And what we see, and we're plotting the RB values in ohm centimeters squared on the vertical axis, we see that when thrombin is added, the barrier function was around 4 ohm centimeters squared, drops to 0 in this case. The model actually drops out of the measurement of this case and comes back, but notice alpha stays constant, suggesting that the thrombin addition is changing the barrier function with little effect on the cleft between the substrate and the basal membrane. Well, that's what I wanted to cover in today's webinar. We offer different instruments. The Z-theta is the workhorse instrument that we've used for most of the data you've seen. There are adapters that go in the incubator that allow you to couple in 96-well or 8-well arrays. And one of the 8-well arrays we are about to re-offer, we had it on the market for some time, is one that allows you to use this instrumentation to look at transmembrane filters and study cells grown in that case and getting tier values. We can study endothelial cells under flow conditions by simply using a special array and pumps where you can see the barrier function of the cells as they're exposed to shear stress similar to that they would see in vivo. Finally, the Tier 24 is also a new instrument that we offer, and you can find more about that on our website, which is biophysics.com. And if you go there and click on barrier function or publications, you can learn a great deal more about this method of monitoring barrier function. So thank you for joining us today. I look forward to hearing from you in the future. Take care.